Hi everybody, welcome back to the next class under reproduction in flowering plants. Today we are going to discuss about the megasporogenesis. Megasporogenesis. So what do you mean by the word megasporogenesis? It is the process of formation of megaspores. It is the process of formation of megaspores. It is the process of formation of megaspores from megaspore mother cell. It is the process of formation of megaspores from megaspore mother cell is called as megasporogenesis. So here megasporogenesis means what? The process of formation of megaspores from megaspore mother cell is called as megasporogenesis. Now here if you look into the ovary, okay, if you look into the ovary, we know that in the ovary there is a structure which we call it as what? The ovule. Okay, there is a structure which we call it as the ovule. So here inside the ovule, now there is the process that is taking place, what? Megasporogenesis. Now we studied that throughout the whole ovule is completely enclosed or filled with cells called as nucellus cells. Okay, the ovule in the beginning it will be completely present with nucellus cells, and these nucellus cells are rich in nutritive material. Reserved food material will be there. So nucellus cells will be containing what? Reserved food material that will be there. Reserved food material will be abundantly present in the nucellus cells. So we are talking about megasporogenesis. How the megaspores are formed? One of the nucellus cells. Now if you are looking at here, these are all the nucellus cells that are there. Okay, these are all the nucellus cells that are there. One of the nucellus cells that is towards the micropyle region. So here this is called as what? Micropyle region. It is the region where the integuments are enclosing the whole ovule but leaving some place or an opening that is present called as micropyle. So towards the micropyle region, one of the nucellus cell will be undergoing the changes. That is very important. So one of the cells at the micropyle region in the ovule is undergoing the changes. That one cell is entirely looking different from all the other nucellus cells. That one cell which will be undergoing the process of megasporogenesis will be looking entirely different from all the other cells of the nucellus. That one single cell we are calling it as what? Megaspore mother cell. This megaspore mother cell is nothing but what? One of the cell of the nucellus present in the ovule. So this one cell is called as megaspore mother cell. So this megaspore mother cell will be containing dense cytoplasm, means what? Very thick cytoplasm, and at the same time a prominent nucleus, means it will be very clearly seen the nucleus. So these are the two characteristics to identify, yes, this is the megaspore mother cell. But what is this megaspore mother cell? Actually it is nothing but one of the cell of the nucellus. It is only present towards what? Micropylar region of the ovule. So the process of formation of megaspores from megaspore mother cell is called as megasporogenesis. Now this megaspore mother cell will be undergoing what type of division is important. That's very important. That megaspore mother cell, okay, in short we can call it as MMC, megaspore mother cell. This megaspore mother cell will undergo meiosis. It will undergo meiosis and produces how many cells it will produce? 4 cells. Means one megaspore mother cell, this is one megaspore mother cell. When it undergoes meiosis, it is producing 4 cells. And from these 4 cells, all these 4 cells are haploid. So all these 4 cells are now called as megaspores. Okay, are called as megaspores. So this is what we have to remember when we talk about the megasporogenesis. So all these points are very important when we talk about the process called as megasporogenesis. Now we are going to discuss about the next topic called as in a detailed way we are going to study how this embryo sac or the female gametophyte is formed. So we just completed about the megasporogenesis. Now we are going to discuss about the female gametophyte. So at the end of megasporogenesis, we said that there are 4 cells that are formed. So which are formed from megaspore mother cell. So if we assume that this is a megaspore mother cell, when it has undergone meiotic division, meiosis, so it has formed 4 cells. It has formed 4 cells, and all these 4 cells we are calling them as megaspores. And from these megaspores, okay, from these 4 megaspores, only, okay, in majority of the flowering plants, 3 will be degenerating. These 3 will be degenerating, and only 1 will be maturing into a female gametophyte. So now let us take that one female gametophyte, which will be called as the embryosac. So this is only one cell is there. The 3 have degenerated. The 3 megaspores degenerated, and only one megaspore is undergoing the growth and development into a female gametophyte. So this is that one female gametophyte which is growing into a female gametophyte, which is the megaspore. So this is having its nucleus, and this megaspore will be undergoing mitotic division. Please remember, here it is meiosis, and here this megaspore, which will be growing and developing into what female gametophyte, will be undergoing mitotic division, mitosis. Will be undergoing mitosis. So what is undergoing mitosis now? First, let us make it very clear. The nucleus inside this megaspore will be undergoing mitosis. The nucleus inside the megaspore will be undergoing the mitosis. Okay, will be undergoing the mitosis. So when the nucleus undergoes mitotic division, here are the cells. So let us take that one cell from there. Here, if this is the cell, when it undergoes first mitotic division, it is forming 2 nuclei. This is the megaspore. It is having the 2 nuclei. Again, these 2 nuclei will undergo another mitotic division, forming 4 nuclei. Again, these 4 nuclei will undergo again repeatedly another mitotic division, forming 4 at one pole and 4 at the other pole. So see here now, if you are seeing one nuclei, it has undergone mitotic division and divided into 2 nuclei. These 2 nuclei move to the opposite poles of this megaspore. So once they moved, now each one at its own pole, it is dividing mitotically. Here, this nuclei divided into 2. And these 2 divided mitotically and formed 4 nuclei. And these 2 divided mitotically and formed another 4 nuclei. They are at opposite poles. And totally there are 8 nuclei in one single cell. Totally there are 8 nuclei in one single cell. And immediately right after the nuclear division, what should take place? There will be cell wall formation. But here we are not seeing cell wall formation immediately after the cell division. Little later, the cell wall formation will be taking place. So that's why whenever these nuclei are there present as freely, we call them as free nuclei. Okay? We call them as free nuclei or free nucleus or nucleus. Okay, let us say free nucleus that is present. Strictly they are free nucleus because they are not immediately enclosed by cell wall. Okay, cell wall formation is not seen. And little later on, you will be seeing that there will be formation of the cell wall around the nucleus. But here, one point we have to remember is that around out of 8 nuclei, there are 6 nuclei that are now covered by the cell wall formation. Means around 6 nuclei there is a cell wall formation. This is first cell, second cell, third cell. And in the same way, at the opposite pole, 4th, 5th and 6th, around these 3 also there will be cell wall formation. But those 2 nuclei that are present at the center, we will not see the cell wall formation. That is important. And now when we see here how these cells are being distributed throughout the embryosac, or otherwise called as the female gametophyte. Please remember, female gametophyte is also called as the embryosac. Okay, female gametophyte is also called as the embryosac. So what we said now, there are totally 8 nuclei. 6 nuclei are now enclosed by the cell wall, and they are now three cells are at one pole, that is at chalazal end. That is at the chalazal end. That is the base of the That is the base of the ovule. That is the base of the ovule. So at the base of the ovule, which is called as chalazal end, we are seeing 3 cells called as antipodals. And another 3 cells which are enclosed by the cell wall are present at the micropylar end. And among these 3, 2 cells which are towards the sides and present at the center will be seeing the egg. So it is like this. Egg will be present at the center, and whereas the 2 cells will be present towards the sides. And these 2 cells which are towards the sides and egg at the center, will be these 2 cells towards the sides will be called as the synergids. And at the center, what is there? Egg is there. But here, these synergids are having beak-like structure, we say. Otherwise, we are calling it with a technical term called as filiform apparatus. We call it as filiform apparatus. The synergids are having the filiform apparatus, through which these filiform apparatus will be directing the pollen tube into the synergids. And we know that when the pollen grains fall on the stigma, they travel through the style, enter into the ovary, enter into the ovule. Entering into the ovule is through the micropyle region. So when they enter, this is the micropyle region. Otherwise, we can say like this. This is the micropyle region now. So when the pollen tube is entering inside into the embryo sac, here they are directed to fertilize with the egg with the help of what? Synergids, because they have the filiform apparatus. So filiform apparatus will direct the pollen tube into the synergids and then to fertilize the egg. And now we are seeing at the center, these 2 nuclei are called as polar nuclei. These 2 nuclei are called as the polar nuclei. And just now we said 3 are present at the chalazal end and 3 are present at the micropylar end. And when you say here, this egg and synergids, these two together we call it as egg apparatus. We are calling it as. And now what is left over? Another 2 nuclei that are left over here. There are 2 nuclei that are placed just behind, or in this diagram it is like just above what? The egg apparatus. So here there are 2 which are called as what? Polar nuclei. So these 2 polar nuclei are present at the center, but they are not enclosed by the cell wall. And where are they present means this whole cell. Now this whole cell is called as what now? The central cell, we call it as central cell. So if you are looking at here now, while we are ending up this, if you look at, we have to count how many nuclei are there and how many cells are present. So if you are counting now the number of nucleus, 1 here, 2 second nucleus, 3 third nucleus, 4, 5, 6, 7 and 8 nuclei are there. So 8 nucleic stage this is. 8 nucleus are there. But when you count the number of cells, it will be only 7. So here what we said just now, we counted 8 nuclei. And now if you are counting the number of cells, 1, that is 1 synergy, second synergy, third is egg, fourth is antibodal, fifth is antibodal, sixth is antibodal. There are 3 antibodals, 6, 3 here antibodals, 3 here as egg apparatus, total 6, and 1 more cell is the central cell. So here are 3 and here are egg apparatus consists of 3 cells, 3 plus 3 6, and the last one is the central cell. So totally how many cells are there? There are about 7 cells. This is very, very important. So in a female gametophyte, which is so called as embryosac, it will be having 8 nuclei and 7 cells. This is how the process of formation of the female gametophyte will be taking place, and the distribution of the nuclei will be taking place.